Okay, so hello everyone. My name is Alex Fornito and I'm from Monash University in Melbourne, Australia. And I'm really sorry that I can't be there in person with you all. Would have been really excited to be back in person at OHBM, but unfortunately I wasn't able to make it. So hopefully this virtual talk will do. Really as an introduction to this educational workshop on network neuroscience. So throughout the day, you're going to hear about a lot of different topics, both basic, fundamental, and applied and advanced topics about how you might use the tools of network neuroscience in the analysis of brain imaging data. And my talk today is really just to serve as a gentle introduction to some of the basic concepts that will serve as a foundation for the other topics that are covered in the other sessions. So we'll begin with trying to motivate why you might want to use network techniques to understand the brain. And it really boils down to this idea that there are really two fundamental pillars or principles of brain organization. And that's functional segregation and integration. And ideally, we want to understand how both of those are implemented and instantiated and emerge in the brain. And really, historically, neuroscience has spent a lot of time trying to understand functional segregation. So this can be traced back to Franz Joseph Gall's work in the 1700s, where he thought that different parts of the brain were specialized for performing different functions, and that the degree to which a particular ability or trait was important in someone's character was related to the size of that area. And so you could go around and measure bumps and protrusions on the scalp, and that would give you an indication of the underlying size of the area. And then you could generate a map that looks like this, where he localized different abilities to different parts of the brain based on his measurements across people from all sorts of different walks of life. Now we know this is pseudoscience, but that basic idea that different parts of the brain are specialized for performing different processes or functions has certainly been a really powerful concept in neuroscience in the time since. So if you fast forward to the middle of last century, we had maps like this that were in circulation. You can see that the sophistication of the terms has improved, and we've got a much more precise mapping. This is based on the collation of a number of physiological and lesion studies. But the basic idea is the same, that different parts of the brain are more or less specialized to perform different roles. And certainly this has been a real fundamental assumption in a lot of human brain imaging. And so if we look more recently, our techniques are advanced to the point where we can look at maps of functional segregation and specialization at a really fine scale. And so this was a study done a few years ago now, where they were able to map regions of the brain that were involved in processing individual words in natural speech. So our ability to localize segregated and specialized functions in the brain is quite advanced. And by comparison, historically, our understanding of the processes that support functional integration has lagged behind. So this is a map of the connectivity between different areas comprising the macaque visual system that was published by Dan Fallerman and David Van Essen in 1991. It's a real landmark paper. They were able to generate this through collating the results of many different track tracing studies. And shortly afterwards, Francis Crick of DNA fame and the neuroanatomist Edward Jones published this essay in Nature, praising Fallerman and Van Essen's work, but really lamenting the fact that we had no comparable map of brain connectivity for the human brain, and really arguing that it's the connectivity between brain regions, the integrative processes that underlie coherent behavior, and we just had no understanding of how that happened in the human brain. It was an auspicious time for them to be writing this, because just a year earlier, we had the discovery of the bold contrast. And in that essay, they actually note that this new contrast had been discovered, and perhaps that might provide a useful way forward. And shortly afterwards, we also had the development of diffusion tractography, which really revolutionized our ability to generate maps of anatomical connections between brain regions non-invasively. So if you then fast forward to 2005, the techniques had advanced to the point where we had Olaf Sporns, Giulio Tononi, and Rolf Kotter in this key paper calling for a map of the human connectome. And they define the connectome as a comprehensive structural description of the network of elements and connections forming the human brain. So the two key ingredients of this definition is a structural description. So we're really talking about the physical or anatomical links between brain regions, essentially a wiring diagram. The fact that there's no specific scale at which this is defined. And this is really because we find networks at all different scales in the brain, from networks of molecules interacting at the synapse through to networks of individual neurons and synapses and populations of neurons linked by axonal fibers. And then at the macro scales that we typically deal with human brain imaging, large macroscopic areas linked by bundles of axonal projections. At each of these levels, we can map a connectome representing brain connectivity at a different scale. There's really no privileged scale of analysis. Each scale gives us different, interesting, unique information. And a real challenge for the field is trying to understand how the different scales relate to each other. Most of what you'll hear about today will largely focus on the macro scale, because that's essentially where most of human brain mapping is limited to because of resolution limits on our imaging techniques. Now, a key aspect of defining a connectome is viewing or modeling the brain as a network. And networks are generally modeled as graphs of nodes connected by edges. Nodes represent the fundamental processing units of the brain. And so this could be individual neurons or populations of neurons, and edges represent the interactions between nodes. So it could be some aspect of anatomical connectivity or functional connectivity or the like. Now, there's also some cases where we can treat edges as nodes in a network or a graph. And I believe Rick Betzel will be talking about this a little bit later. But intuitively, as a base, we think about nodes representing brain regions and edges, the interactions between them. Now, this is a very general representation of the network, this graph-based representation. It's used to represent diverse networks found right throughout nature, from protein interaction networks to social networks, technological networks like the World Wide Web, and also transportation networks such as train lines or global air transportation networks. In each of these cases, the units or elements are the nodes and the links between them are the edges. But the interpretation obviously varies depending on the context. Now, the nice thing about this general way of representing networks is that not only does it provide us a lot of flexibility, but it also allows us to relate our understanding of brain networks to a much broader literature of network science, where there's been a lot of work going into the physics of networks, a lot of different models. And so we can contextualize the brain within a much broader web of different naturally occurring phenomena. And we can ask, well, is the brain similar or different to these other types of networks that we find in nature? So the core ingredient then is really how we define our nodes and edges. Everything that happens after that is going to be dependent or contingent on these decisions. And particularly with human brain imaging, it's not so clear cut. If we were looking at networks at the micro scale, we could say, okay, well, each neuron represents a node, and the connections between them are some synaptic connection or some index of coupled activity. But at the macro scales that we deal with human brain imaging, it's not so clear cut. So I'm going to talk a little bit about how we define nodes. Because of the ambiguities in how we can do that at macro scales, there's a few different criteria or approaches that have been proposed, and we generally use heuristic methods. Generally, though, a node should ideally be spatially constrained. We typically think of a processing unit of the brain as something that is anatomically constrained, so an anatomically contiguous patch of cortex or subcortical nucleus, and that's intrinsically homogeneous, meaning that all the constituents of that area will share some element of function. So, for example, if we're clustering voxels together, we expect those voxels to have a similar function. And they should be extrinsically distinct. So the functions of one area should be distinct from the functions of another area. Now, the classic way of parcellating the brain was Brodmann's cytoarchitectonic map, and there have been attempts to generate probabilistic cytoarchitectonic maps that are mapped into stereotactic space, although presently these do not extend to cover the entire cortex. There's alternative techniques or criteria. One could use chemoarchitecture, myeloarchitecture. People have used sulcal and gyral anatomy. People have just defined parcellations at random at various scales. People use different functional criteria to define their nodes. There's a whole area looking at data-driven parcellations, so using various clustering techniques to group voxels or surface vertices together with similar activity. People have used individual voxels as different nodes. And a few years ago, there was a landmark paper from the Human Connectome Project using a multimodal approach to try to identify key areas of the brain. Now, each of these different approaches has different strengths and weaknesses, and it really depends on your application, how you'd like to interpret it. But you need to be wary of the fact that the way in which you define your nodes will constrain interpretation. And there are also more technical or pragmatic aspects about these choices that affect network analysis. So for example, if you have a parcellation that has regions that vary a lot in size, so say for example, this multimodal parcellation here, you can see areas of somatosensory and motor cortex are very large compared to parcels in prefrontal cortex. That can affect or bias certain estimates. So for example, if we're just trying to look at the overall connectivity of one of these areas in, say, a connectome mapped with diffusion tractography, then larger areas are going to have more streamlines attached to them, and they'll have a higher degree of connectivity simply because they're larger. And so you need to be aware of these potential biases and address them in your analysis. So that's definition of network nodes. Now I'm going to talk about how we define network edges. And so really the edges are trying to map connectivity between nodes. And there's three broad classes of connectivity that have been defined in the brain. There's structural connectivity, which is the physical or anatomical connections between brain regions. Now this is intrinsically directed. Each axon has a source and projects to a target, but we can't resolve that directionality with MRI. So the maps that we typically obtain with MRI are undirected, and that's just a limitation of present technologies. Then there's functional connectivity, which is a statistical dependence between physiological signals that we record in different areas. So it's critical to remember this is estimate the level of measured signals, and it can be directed or undirected. So most commonly it's estimated with a correlation coefficient, which is undirected, but measures such as say Granger causality applied to physiological signals are best thought of as measures of directed functional connectivity, and they can be distinguished from effective connectivity. So effective connectivity is the influence that one neuronal system exerts over another. And so really we're trying to make inferences at the level of neuronal populations, not measured signals. And this typically involves some model that relates the underlying neuronal dynamics to the signals that we measure. So this in the imaging literature is perhaps most famously exemplified by dynamic causal modeling, where we specify different models of neuronal interactions, and then there's a forward model that maps those neuronal interactions to measured bold signals, and we work out which model best explains the data. And so that's why effective connectivity should be distinguished from directed functional connectivity. So directed functional connectivity is purely looking at causal interactions between measured signals. Effective connectivity is trying to understand the underlying hidden neuronal states that are driving those signals. And so this distinction is quite important when we use very indirect measures of brain activity, such as the bold response. If we are using more direct measures, say we're looking at interactions between LFPs or spiking neurons, then we can more readily apply techniques such as Granger causality as a more direct index of effective connectivity. So there's many different approaches and techniques out there for mapping both structural and functional connectivity in the literature. I'm really just going to focus on those that are used most commonly in human neuroimaging. So that's diffusion MRI and functional MRI. So we'll start by just talking about one of the key aspects when generating these connectomes. And that's the fact that whether you use diffusion MRI or functional MRI, the connectomes that you generate rely on a complex sequence of data acquisition and processing techniques. So we typically acquire the data in the scanner, and we have lots of different choices in terms of the acquisition parameters that we can use. For example, in fMRI, you could choose to use multiband or not multiband, multi-echo or not multi-echo. You can play around with the different temporal and spatial acquisition parameters. With diffusion MRI, again, you could choose multi-shell or single shell. You need to determine the number of directions, the number of B values, again, spatial and temporal, spatial resolution factor, spatial resolution parameters, and so on. Then we acquire those data and we pass them through some processing pipeline designed to extract the information we want and minimize noise as much as possible. And then we build a connectome map. Now, the data, this step in the middle here, the data processing pipeline is really critical. And in both functional and diffusion imaging, these processing pipelines are very complicated. They involve lots of steps, and at each step we need to make lots of different choices. And the choices we make often affect the final outcome. So, for example, this is one study we did a little while ago, looking at the effects of different techniques for addressing motion-related confounds in functional connectivity analysis with fMRI. We looked at a range of different pipelines here, represented along the rows here of this graph. And these bar charts represent the degree of residual motion contamination after denoising with each of these pipelines. And you can see there's quite a lot of variability here. In this case, we found for some pipelines, almost 70% of connections showed some level of residual motion contamination, compared to other pipelines down to 6%. Right, so the choice of pipeline really does have a big impact on your final connectivity estimates, and it does have a practical impact on the results. So for example, here we looked at the effect or the conclusions you would draw from a case control comparison of people with schizophrenia and healthy controls. And here we plot the proportion of significantly different connections, either in one direction or the other. And a whole series of pipelines found no differences. Some pipelines did find differences. And so again, the pipeline you apply is going to strongly affect the conclusions that you reach. And again, this is just showing the distribution of the connections that differed between the groups for two cases where we did find differences. So we've got two different processing pipelines at the top and the bottom, connections that were increased in patients versus decreased in patients on the left and right respectively. And you can see there's, the specific networks that are affected do vary depending on the pipeline that is being used. So these choices are important. It's important to understand the choices and to consider their effect on your analyses. We don't have time to go into a detailed consideration of these different steps, but for those who are interested, there's some recommended reading down there. A diffusion MRI is not immune to this. Again, this is a study we did a little while ago where we again found that different pipelines varied considerably in the degree of motion-related contamination. Although in this particular case, we found that the use of the latest version of FSL's EDDI to deal with motion correction as well as outliers and some other artifacts does a pretty good consistent job of mitigating motion-related confound. And of course, head motion isn't the only problem. Particularly in fMRI, we have all sorts of different sources of physiological noise. One that's attracted a lot of recent attention is the role of fluctuations in respiration depth or volume. And this slide just shows a trace from a respiratory belt showing how the person's breathing varies over time. And you can see that these low frequency variations in respiration depth track quite nicely these global shifts in signal intensity. So in this plot here, each row is a voxel, each column is a point in time, and the color indicates the signal intensity. You have these brain-wide shifts in signal intensity that correspond quite nicely with changes in respiration depth. And so obviously any correlation estimates you obtain from these data are going to be strongly dominated by these large scale shifts. Actually, and this study by Jonathan Power a little while ago showed that acquiring these respiratory traces and correcting for them doesn't fully remove these shifts. And in this particular case, advocated for using a tool, a technique such as global signal regression. There are other alternatives. Again, we won't go into the global signal regression debate, but there are some articles down there for interested readers so they can weigh up the pros and cons of that specific denoising procedure. Right. Now, assuming that we've navigated those issues and we're happy with our processing pipeline, our next step is to then build a connectome. And there's a pretty well-established pipeline for doing that. We divide the brain into different areas that act as our network nodes. And then for every pair of areas, we measure some aspect of structural or functional connectivity between them. And we represent that in the form of a connectivity matrix, sometimes also called an adjacency matrix. So here, each row and column represents a different region, and each element corresponds to the type and connectivity between pairs of regions. And with this matrix equivalently, we can generate a graph-based representation of the network where we've got the locations of our nodes and we draw an edge between them if there's a connection between them in the matrix. So I'm just going to focus a little bit more on the properties of the adjacency matrix. So you can see here, this is just a sort of zoomed in version. And so, for example, here, this element 1, 2 tells us about the connectivity between region 1 and 2. It's quite strong. Between region 1 and 3, it's moderate. Between region 1 and n, it's weaker. In this particular case, the matrix is symmetric. The diagonal tells us about the connectivity of each region with itself. And so in this case, it's a correlation network. So we just have 1. And you can see that the value for element 1, 2 is the same as the value for element 2, 1. So because it's a correlation matrix, the connectivity is undirected and the matrix is symmetric. So more generally, we can say that the upper triangle of the matrix, all the elements above the diagonal, are the same as the lower triangle of the matrix, all the elements below the diagonal. And so in an undirected network, we generally say that the element Aij is equal to Aji. In a directed network, say we've looked at effective connectivity or we have track tracing data and we can resolve directionality of connectivity, the upper and lower triangles are not necessarily the same and connectivity is asymmetric. So here we have Aij does not equal Aji. Now, that implies that we can generate different types of graphs from our adjacency matrix. So here is the simplest case. We've got a binary undirected network. So it's an undirected network. So again, the upper and lower triangles are symmetric and it's binary. So we're not incorporating any information about variations in connectivity weight. We're just asking where connections are in the network. And so here, this is the graph equivalent of this matrix here. So for example, here you can see there's a connection between node A and C. And we draw a line between C and A and C, and also between A and E. And again, you see a line between A and E. And equivalently, if we go, there's a connection between E and A, and between C and A. Right? So the first row is the same as the first column. Now, assume we want to look at variations in connectivity weight. We know that they vary over several orders of magnitude, so it's quite important to account for that information where possible. So in this case, here we've got weights. The colors represent variations in connectivity strength, and we can represent that in our graph by just varying edge thickness. So you can see that the connection between node C and F is quite strong at a value of 20, so that's a lot thicker than, say, the connection between C and A, which is a value of 13. Then we also have directed networks. So in this case, we have a binary directed network. So you can see here that we have a connection going from A to E, but not vice versa. And in this particular case, we list outgoing connections down the columns. That choice is arbitrary. We're just using that convention here. So here, down column A, we see a connection to node E, but down column E, there's no connection to node A, right? So in this case, the matrix is asymmetric, representing directionality in the network. And then we can do the same thing if it was weighted. We've got the same basic information, we just vary the weights on the connections and the thickness of the edges. So that's, that's some basic varieties, but we can use more complex graph representations of the network. And so we can ask, well, what type of network best represents the brain? So we've got the simple case. We say, okay, well, we want to map a brain network. So we find our nodes and we map which pairs are connected. But as I mentioned, those connections typically vary over quite a wide range in terms of their strength. So maybe we want to represent that variation in connectivity strength. So we need a weighted network. I've also mentioned that connectivity is intrinsically directed. So ideally, we would resolve that directionality where possible. And we also know that different brain regions are not the same. So they're heterogeneous in terms of their function, their cytoarchitecture, their molecular properties. And so we might want to represent that in our network model in some way and try to account for that variability and see how it impacts dynamics. And so ideally, we would incorporate node heterogeneity. And we also know that edges are not the same either. Some are inhibitory, some are excitatory. And so again, we would ideally represent that. And so really, the dream representation would be some kind of hybrid graph that accounts for edge and node heterogeneity, directionality, and weightedness as well. And it would be also something that varies through time, because we know dynamically brain networks vary on very rapid scales, and structurally they vary over scales ranging from days to months to years. Most studies in the field still rely on binary undirected representations. An increasing number are using undirected weighted representations. But really, particularly with imaging, directionality is a challenge, certainly with the availability of comprehensive track tracing data sets in animals. We're starting to see more analyses of directionality, but it is a challenge with human imaging. And some studies are starting to look at edge heterogeneity. Some people are also beginning to look at node heterogeneity, but it is early days. There is evidence that accounting for this heterogeneity can certainly improve, say, dynamical models of brain function. So the bad news is that our network models are quite rudimentary, but the good news is that this approach is sufficiently flexible that it can accommodate technological advances, so that we still have a lot of room to grow and improve in the sophistication of our models. But assuming we're happy with our model, we then want to analyze our graph. And really there's two broad classes of analysis we can do. So one focuses on connectivity. So this is looking at variations in the strength of connectivity between pairs of nodes. And the other focuses on topology. So topology is really concerned with the organizational property or the arrangement of connections between pairs of nodes. It's really any property of the network that is invariant to a continuous spatial transformation of the network. So you can imagine if we took this network here and rotated it or stretched it, the pattern of pairwise connections would be the same, and that defines the topology of the network. And so this is really where we use the mathematics of graph theory to derive a whole range of different metrics that characterize how the network is organized. And for the other talks today, you're going to hear different varieties of these different connectivity and topological analyses. So to summarize, hopefully I've convinced you that graphs can provide a useful model for brain networks, and really a single framework for understanding multi-scale organization in the brain. There's different ways of defining brain nodes and edges. There's no single gold standard. Each approach has its advantages and disadvantages. And it's important to be aware of those, because this will constrain the way we interpret our findings. Brain networks can be direct or undirected, weighted or unweighted. We can also account for node and edge heterogeneity if that's a goal. And brain networks can be analyzed at the level of either connectivity or topology, and we can map effects at the level of individual edges, nodes, or more globally, as you'll begin to hear about in the other talks. So I'll just wrap up by pointing you into the direction of some resources. Here are some general introductory texts on network science. Here's some more specific texts on network neuroscience in particular, and articles. A few years ago, Andrew Zaleski, Ed Bullmore, and I released a textbook that covers a range of different network approaches to analyzing neuroscience data. And we also have a journal with Olive Spawns as editor-in-chief, Network Neuroscience. It's open access, very competitive publication charges. So if you are working in this space, please consider sending your work there. And I've also put some links here to some different toolboxes, some of which you'll hear about later today. But that brings me to the end of the talk. So thank you very much for your attention. And I'll hand over to Andrew, who will hopefully be able to field all of your questions. Thank you.